Welcome to the second and final episode of the Mesenchymal stem cell mini-series. In case you haven't watched the first episode, mesenchymal stem cells, or MSCs for short, are one of the most debated topics in the stem cell field. In this episode, we're going to talk about the science behind how MSCs work, clinical trials using them, and what the future of the field might look like. If you haven't watched the first episode yet, which was all about the debates around MSCs, make sure to click right here to watch it and then come back. Don't worry, I'll wait. Okay, enough waiting, let's dive in. MSCs actually started their journey as feeder cells, cells that are used at the bottom of petri dishes to support the cells on top, which are usually the important cells that scientists are actually studying. In the case of MSCs, they were being used to support hematopoietic or blood stem cells. However, it's important to note that those feeder cells weren't necessarily being characterized the exact same way we characterize MSCs today. Nowadays, MSCs have generated lots of scientific interest. They've really turned things around for themselves, but their rise to the top has not been without controversy, as we saw in the last episode. MSCs were first described and called mesenchymal stem cells in 1991 by Kaplan, who we also talked about in the last episode. But by the early 2000s, the apple and peach dilemma had arisen, and scientists weren't always talking about the same thing when they said MSCs. I find it helpful and interesting to think of the history of the MSC field as a long-running TV show. At the beginning of the show, the protagonist was called mesenchymal stem cell and had some pretty distinct characteristics. But over time, new writers began writing for the show, and what was canon or not about this character became unclear. In fact, the show became more of a sci-fi rather than a documentary. But now, it seems like the show is regaining its footing and getting grounded in reality once again, though some scientists might disagree with this. It seems like the line between what's canon and fan fiction is becoming clearer. Moving back to reality and away from the fictional world of the TV show, it's time to return to the apple and peach dilemma, but a slight variation of it. Remember that with the apple and peach dilemma, scientists may be referring to both apples and peaches as apples, which clearly makes things confusing. But another issue is that scientists may think a population of cells is homogeneous when in fact there's a lot of diversity within it, and then they'll be calling those subpopulations by the same name. So for instance, we could have a group of oranges, pears, and apples, and not know that they're different and just call them all apples. The best example of this comes from the source of MSCs. Though MSCs were initially found in the bone marrow, scientists soon began discovering them in other tissues as well, such as adipose or fat tissue. At first, scientists were grouping all of these MSCs from different tissues together and calling all of them MSCs. But scientists now know that the source of the MSCs matters a lot and can affect the function of the MSCs. So the ISCT has asked that scientists include the tissue source when writing about MSCs. In a 2016 paper published in Stem Cell Reports, Benedetto Sacchetti and colleagues found that MSCs from different tissues had different gene expression and different capacities to differentiate. In one part of the study, the scientists looked at how MSCs from various sources worked in vivo. Remember, that means inside the body. Looking at muscle formation in mice, for example, the researchers found that skeletal muscle-derived MSCs contributed to injured muscle, while non-skeletal muscle-derived MSCs, like MSCs from the bone marrow, didn't. Looking at this, it becomes pretty clear why not distinguishing between these different cell types can lead to major setbacks in the field and even be potentially harmful. Other scientists, like Dragomir Jovic and colleagues, have found that even within the same population of MSCs derived from the same tissue, there can be diversity. Other sources of heterogeneity of MSCs, apart from being derived from different tissues, include how the cells are frozen, which is done to preserve the cells, how they're thawed, how they're cultured or grown in the lab, and how they're isolated. Isolation is the process of taking bone marrow, as an example, which has many different cell types, and then isolating just the MSCs. Despite all of this confusion, there have been successes within the MSC field too. For example, it's now well established that MSCs can be helpful for treating graft versus host disease. Though there are some varying reports on this, the results are generally positive. I'll turn it to Dr. Horwitz, who we met in the first episode, to explain what graft versus host disease is and how MSCs are thought to help with it.
Because MSCs were first described as modulating the immune system in 2001, I believe it was. So what seems to happen in graft versus host disease is after bone marrow transplant, the donor's T cells, which are part of the graft, attack the patient's body, and the patient is the host. So it's graft versus host. And what you need to do to treat it is to suppress those immune cells attacking. So you need to suppress the immune attack. And MSCs can do that. And exactly how they do it is still a point of contention. It's not clear exactly what they're doing. And it seems to me that it doesn't have to be one mechanism. There could be multiple mechanisms.
Dr. Horwitz himself also pioneered the use of MSCs for osteogenesis imperfecta, or what's commonly known as brittle bone disease. Brittle bone disease is genetic and leads to weak bones, fractures, and shorter height, as well as other bone-related issues. Now back to Dr. Horwitz. So you mentioned the 2002 paper about osteogenesis imperfecta, and so could you talk a little bit about the proposed mechanism of action, about how MSCs could be helping with that?
Well, yes. So I'll give you two answers. At the time, it turns out this isn't true, but at the time, we believed that the MSCs were osteoprogenitors. Whether they were stem cells or not, weren't commenting on, but they're osteoprogenitors. And in fact, osteoprogenitors mean they are progenitor cells that give rise to bone. And in my 1999 paper, the title is mesenchymal cells. I don't actually say mesenchymal stem cells because I thought the data to support that statement was lacking. So I didn't want to be on the record as calling them stem cells. So I didn't. And the 2002 paper, again, we call them mesenchymal cells. And the thought at the time was they were going to engraft in bone to give rise to new osteoblasts, healthy osteoblasts. Then what happened was, at the time, we were allowed to do gene marking, and we found very low levels of engraftment in the bone. So then we were trying to explain how such low levels could actually have such striking clinical benefit. The benefit was amazing, but the engraftment was very, very small.
We'll really explore clinical trials in the next chapter, but for now, let's shift gears towards MSC's paracrine signaling and what they secrete that results in the positive effects we've heard about. Since MSCs themselves have been found to die quite quickly in the body after having been injected, scientists think MSCs work through a hit-and-run mechanism where they secrete the beneficial factors and then die. This hit-and-run analogy was used by James A. Ankrum and colleagues, by the way. The factors MSCs secrete are collectively called the MSC secretome. But what exactly is this? A mixture of different biologically active molecules, including, but not limited to, growth factors, cytokines, chemokines, interleukins, microRNAs, and extracellular vesicles. Before you click away, don't worry, I'm going to explain what all of these terms mean using an analogy of a town called MSC Town. First up, growth factors. They're the grocery store of the town, as they help the residents grow. In reality, growth factors help cells grow, helping with both division and differentiation. Up next, cytokines. They're the biggest branch of a chain of stores called cytokines, and they offer all the services and products that the other branches offer. In the biological world, cytokines are a large group of pro- and anti-inflammatory factors. Next up, chemokines. They're a smaller branch of the big cytokines chain of stores, specializing in helping residents move. In the biology world, chemokines are a subgroup of cytokines, helping specifically with cell migration. The next term is interleukins. Similar to chemokines, interleukins are a branch of the big cytokines chain, specializing in moving, career counseling, and more. In the world of biology, interleukins are a subgroup of cytokines, are pro- or anti-inflammatory, and help with cell migration, differentiation of immune cells, that was like career counseling, and more. The next term is microRNAs. They're the sheriff of the town, making sure the residents stay in line. In reality, microRNAs don't code for proteins and instead negatively regulate gene expression, meaning they make sure certain genes aren't expressed. Finally, we have extracellular vesicles, which are the taxis or Ubers of the town, helping residents get around. In our bodies, extracellular vesicles are tiny sacs that transport other biological factors like proteins, DNA, microRNA, and more. So what can these factors actually do that might help with diseases? There are a lot of different claims as to what the MSC secretome can do, but some of the most widely agreed upon ones include being anti-inflammatory, immunomodulatory, which means changing the immune system in some way or affecting it, and angiogenic, meaning causing the formation of new blood vessels. Also, being anti-inflammatory and immunomodulatory are closely linked, since the immune system can cause inflammation. Let's dive into a specific example. One molecule of microRNA, called microRNA 21, which has been found in the MSC secretome, has been shown to help with heart attacks through helping with inflammation, better functioning of the heart, and better remodeling of the heart after heart attack. To better understand the role of microRNA 21, specifically in heart attacks, it's time to go into the lab. When Lin Shan Yang and colleagues reduced the amount of microRNA 21 compared to normal levels found in mouse models, they saw there was greater cell death of cardiomyocytes, the muscle cells of the heart, after heart attacks. And these microRNA 21 deficient mice had higher mortality than mice with normal levels of microRNA 21. When microRNA 21 was knocked out, meaning there was no microRNA 21 present, there were higher levels of inflammatory cytokines, leading scientists to believe that microRNA 21 may be lowering the production of inflammatory cytokines, leading to less inflammation and possibly lower mortality. Pretty crazy how a tiny molecule can be so important. Other studies have found that components of the secretome can minimize fibrotic scarring after a heart attack, which is when the tissue that's damaged is replaced by a scar that can lead to stiffening of the tissue and dysfunction of the heart. Another fascinating use of MSCs is for delivering cancer drugs, like incredibly helpful Trojan horses, an analogy used by Abdolkarim Hamacha and colleagues in a paper. There is some evidence that when injected into the body, MSCs migrate to sites of injury or tumors, and this is called homing. But this idea is hotly debated and is rejected by some scientists. As well, there's some evidence that in metastatic cancers, MSCs follow cancer stem cells that are migrating to a new location. To learn more about cancer stem cells, check out my previous video on them right over there. In a study exploring this concept, scientists engineered MSCs to contain a type of interleukin that affects metastasis. Remember from our analogy that interleukins help with moving and career counseling. They then injected mice with certain types of melanoma, breast cancer, and hepatoma with these MSCs over 20 days, and they found that the MSCs helped inhibit metastasis progression in these models. But just when you think there's clear evidence of something in the MSC field, confusion strikes again. Researchers have actually found that MSCs can have both anti- and pro-tumor effects. This could be because of the heterogeneity we already discussed, as well as the dose of the MSCs, the timing of the injection, other experimental conditions, or something else entirely that we don't know yet. Despite all of the confusion, there are some other areas MSCs have been found to help with, like wound healing through increased angiogenesis and cell division, faster wound closure, and lower inflammation. In one specific study of mice with cutaneous, that's a fancy word for skin, wounds, the secretome of MSCs from fat tissue led to faster wound healing, minimal scar formation, and higher epidermal/dermal thickness around the wound, which is desirable. One final topic we'll look at in this chapter is the immune system, and there is a lot of research on how MSCs affect the immune system. For instance, one study found that engineered MSCs can increase the survival of mice with glioblastoma, a type of brain tumor, by promoting T-cell invasion — T cells are a type of immune cell — and stopping the division of tumor cells. But as is the case with many topics in the field of MSCs, the immunomodulatory effects of these cells isn't so clear-cut. In fact, it's been found that MSCs can affect inflammation both positively and negatively, like a double-edged sword. Interestingly, scientists think that a certain level of inflammation may be needed to activate the immunosuppressive abilities of MSCs. If this hypothesis is correct, MSCs are like a superhero that only saves the day when things get really bad. Putting it more scientifically, if there's little inflammation in the body, MSCs may not be anti-inflammatory at all. Okay, so it looks like the secretome is pretty great. But one question that popped into my mind as I was researching this topic was, why don't scientists just inject the secretome into patients? Well, as we've already seen with some of the studies we've discussed, the secretome has been used on its own before, and it's been shown to have some positive effects. In fact, using extracellular vesicles alone — remember those are the taxis or Ubers of the town — may make it easier to obtain regulatory approval because it's not a cellular therapy, and they may have a lower chance of causing an immune reaction. As well, compared to single growth factors or cytokines, extracellular vesicles have specific ratios of factors to each other, which may contribute to their effectiveness. Before we hear Dr. Fernandes' answer, a quick disclaimer.
Everything said in this video is my own opinion, based on my own empirical evidence and what I've read in literature, and does not reflect that of my institution or the McEwen Stem Cell Institute. I think defining exactly what is in the secretome and in the exosomes would be very beneficial and really beneficial for patient populations. And the reason why I say this is, if you look at kind of where these are being targeted, they're largely musculoskeletal injuries. And in our current medical system, we don't have very great ways to repair or kind of modulate these injuries in the niche. And what we see with injection of the MSCs or platelet-rich plasma, which is likely acting through an exosome mechanism as well, is that you see reduction in inflammation. You see some modulation of the niche that in either animals results in a better immune profile, better mobility, or in humans that we've seen less pain and better quality of life. So there certainly is something that's useful here, but I think especially to make it a product that we can do in the clinic that is approved by the FDA, we really need a newfound push of rigor into the field, defining exactly what's going on, and what doses is it happening, and why is it happening, before we can really get this into prime time, so to speak. Right now, I don't believe there's any MSc clinical trials that have a good future.
More chance to get more free stuff for your point.
There's a lot of early phase trials, but it's hard to say with any confidence that these trials are showing a clear benefit. They show clear safety and they show the prospect of benefit.
Listening to those clips, it might seem like our discussion is done, and everything we've already talked about was pointless. MSCs are either entirely bad or just too hard to research, right? Maybe not quite. Dr. Fernandes' answer to another question I asked really helped put things into perspective and stuck with me. Remember those illegitimate stem cell clinics that I talked about earlier? I asked Dr. Fernandes about the injections that some celebrities and athletes receive from these clinics that are not regulator approved. Many of them claim to see amazing benefits. So what gives?
I wouldn't say that they are useless. I think that's more so throwing out the baby with the bathwater. Then again, I mean, as talking from I'm not a clinician, nor am I giving clinical advice, nor am I in the position to do so. And I think it would be very irresponsible for one to give the clinical advice to go get this therapy. There's a wealth of anecdotal evidence that comes from this athlete population. Thing is, when you're injecting cells that are your own back into yourself, you do not need FDA approval to do something like this. So given that, these are very easy to do in the right space with enough money. So there's a large wealth of people who have done this now and who have reported effects. So I do think there is something happening, and I do think it is in the realm of immune modulation, probably reducing the inflammatory environment that is in these injured sites.
Throwing the baby out with the bathwater. I think that really applies to the MSc field. When the field was in its infancy, there was a lot of exciting research coming out, some of it misleading, which rightfully tainted the field. But that doesn't mean MSCs can't have a future. As we've already seen, there is a lot of debate and controversy around MSCs, and as we will see, MSc clinical trials are not without their faults. But I still wanted to highlight some of them, some successful, some not. Let's start with a treatment from Osiris Therapeutics called Remestemcel-L or Prochymal, which is the brand name. It involves MSCs from the bone marrow and received conditional approval to treat children with steroid refractory acute graft versus host disease in Canada and New Zealand in 2012 and later in Japan. We already know what graft versus host disease is, and the steroid refractory part means that the patient didn't respond to steroids, which are part of the standard treatment for graft versus host disease. Though Prochymal received conditional approval in Canada, which is approval provided Osiris supplied some more evidence, it was never marketed in Canada, since Canada was not going to reimburse Osiris for the treatments. Soon after the conditional approvals in Canada and New Zealand in 2013, Osiris sold its Prochymal assets to another company called Mesoblast. This is where the story gets really interesting. According to a Cell stem cell paper by Jacques Galipeau and Luc Sensebe, Mesoblast may have changed their trial design from what Osiris used in 2009, that wasn't super successful, so that the new Prochymal trial could lead to a more favorable outcome. For instance, they knew that treating graft versus host disease early on led to better results, and that liver and gut graft versus host disease are more responsive. So those were the types of patients that they recruited. They made a few other changes to the trial design, and the trial was ultimately successful. Keep in mind that this is all based on the aforementioned paper and is speculative, so please don't sue me. Also, if it's shown unequivocally that patients with gut and liver graft versus host disease or earlier on in the progression of the disease are helped by this procamal treatment, then that's an amazing win for patients and the company. But it's still good to note that this is something researchers have talked about. On the approval side of things, Mesoblast has attempted to get approval for this treatment from the FDA. After an initial rejection in 2020, they reapplied for approval recently, but were once again rejected. As a next step, they're taking the FDA's suggestion to run another clinical trial. So how is Mesoblast treatment supposed to work? According to the company's website, quote, Ryoncil, which is the new brand name, is believed to have immunomodulatory properties to counteract the inflammatory processes implicated in steroid-refractory acute graft versus host disease by downregulating the production of pro-inflammatory cytokines, increasing production of anti-inflammatory cytokines, and enabling recruitment of naturally occurring anti-inflammatory cells to involve tissues, end quote. That's quite a mouthful, but put more simply, it's thought that cytokines and anti-inflammatory cells help to reduce inflammation in patients. Another trial I'd like to highlight is for osteogenesis imperfecta, commonly called brittle bone disease, which we learned a bit about in the previous chapter. Beginning in 1999, scientists conducted a trial where they performed bone marrow transplants in children with osteogenesis imperfecta. They saw positive results and conducted a similar but more robust trial in 2001. They found higher growth rates and fewer bone fractures in children who were being treated with bone marrow transplants. At the end of 2001, their working hypothesis was that mesenchymal precursor cells, no, they're not calling them MSCs, but they are similar. These cells engraft in the skeleton and form osteoblasts, which are cells that can form bone. In 2002, the researchers tried to narrow in on their hypothesis at the time and treated osteogenesis imperfecta patients with isolated mesenchymal cells, meaning they received a bone marrow transplant followed by 2 infusions of mesenchymal cells. Note that, once again, they are not calling them mesenchymal stem cells because they aren't necessarily stem cells. What the researchers found was amazing. They saw that patients treated with mesenchymal cells had much higher growth velocities, meaning they were growing much faster. You may have noticed that I said their hypothesis at the time. That's because their hypothesis is now different.
And then 10 years later, in 2012, we published a study in mice that showed that the MSCs actually do stimulate bone growth like we thought, but not by engrafting and differentiating to bone cells, but by secreting something. And we show that it's a secretory mechanism that has the benefit. And there seem to be secreting something that generates something else. So what we have factor A is being secreted from the MSCs, and that generates factor B. And then factor B does the work.
There have also been trials that haven't been successful at all. For instance, there was a large multi-center trial done at 33 centers across the US and UK, where patients were treated with bone marrow MSCs to treat acute stroke. Even though the treatment was found to be safe, there was no difference between the treatment and placebo groups when looking at stroke recovery. As for some earlier phase trials, in a phase 1 trial, which is smaller and focuses mainly on safety, MSCs have been engineered to secrete interferon beta in women with advanced refractory epithelial ovarian cancer. This is a type of ovarian cancer where the cancer has become resistant to treatment. And interferon beta is a cytokine that's been shown to help with various cancers. Scientists found that the treatment was safe and saw homing and incorporation of MSCs at the tumor site. But further trials are needed to see whether MSCs are truly helpful in this scenario. MSCs are controversial. I think that's one thing this mini-series has shown. But the field has also come a long way. Looking at this timeline, which is by no means exhaustive, we can see that cells that could differentiate into bone in the bone marrow were first discovered in the 1970s, setting the scene for the MSC field. 2 decades later, Kaplan wrote his paper titled MSCs, bringing the term mesenchymal stem cells into existence. Since then, MSCs have been used to help with hematopoietic recovery following high-dose chemotherapy, and MSC treatments have been approved in several countries. But some scientists have also made claims now thought to be untrue, such as the claim that MSCs can differentiate into functional neurons. There have been successes and there have been failures. But the past is the past, so what does the future hold? Let's see what the experts have to say.
I think the most beautiful thing is if we can define the perfect cocktail of exosomes. They have mRNA XYB, factor XYB, that's defined in a certain ratio and a dose. We know the dose responses, all the drug kinetic studies, and then that is what comes out of the MSC field. I think the MSC field would have done something great for medicine, because there is a space for those kind of therapies to help people earnestly.
I believe that we will be developing products that MSCs are highly effective to treat. And I truly believe that. We haven't had a major setback in terms of MSCs have never created cancer, which is always a worry. We infuse a cell, will it transform into a cancer? It's never happened. No one's ever died of an MSC infusion. And no one's even been hurt by an MSC infusion. These are all very positives, allowing us to get into more clinical trials. So I think that at some point in the not too distant future, we will get approval for the first MSC product to treat a disease.
The MSC field isn't perfect, not by a long shot. And MSCs aren't a panacea that can suddenly erase every disease off the face of the planet. But scientists are working hard to see what they can do. A huge thank you to Ian Fernandes and Edwin Horwitz for taking the time to sit down with me and share their wisdom. If you'd like to watch the 2 interviews in full, you can click on the links in the description box down below. And thank you to my sister for helping me film. As always, you can find all my references in the description box as well. Finally, if you're ever in doubt about the validity of a stem cell treatment or trial that you or a loved one is thinking of receiving, please consult the International Society for Stem Cell Research's Patient Handbook, also linked down below. Thank you so much for watching. Bye!